To help us wade through this very technical subject, Dr. Kevin Anderson joins me now via Skype. Dr. Anderson received his PhD in microbiology from Kansas State University, was a National Institutes of Health postdoctoral fellow at the University of Illinois, and later a professor of microbiology at Mississippi State University. He taught molecular genetics and served as the major professor for several doctoral and master's students, as well as research microbiologist for the U.S. Department of Agriculture, and is now present director of the Van Andel Creation Research Center in Chino Valley, Arizona. Dr. Anderson has over 20 papers under his belt in technical microbiology and has presented papers at multiple national and international scientific meetings. He's also currently the editor-in-chief for the Creation Research Society Quarterly. Welcome to the show, Dr. Anderson.
Thank you. Thank you for having me.
If you don't mind, I'd like to start off with the special request that sort of prompted this interview from one of our viewers. Stephen wrote in from Virginia. Was wondering if you might find the time to do a piece on molecular clocks on Genesis Week. I mention it now because Evos are bringing it to the table more often than they used to in online debates, whereas before they seem to have virtually discarded it. One of the subjects they are bringing it to bear on is plant evolution. One of the worst places for budding evolutionists to start. There's virtually nothing in the fossil record for them to interpret as transition between the major groupings of plants, whereas there is material for them to misinterpret in the animal record. Anyways, an in-depth look at this would be helpful. Now, of course, most of the genetic clocks have nothing to do with plants, but could you please explain to the viewers just what is a molecular clock? Let's start with that.
Yeah, well, I like your statement that in a sense the plants are so unknown that it gives a lot of room for all kinds of wild ideas, and I think that's why molecular clocks are becoming popular in plants, is because you can make all kinds of statements, because genetics in plants is just so strange that anything you say probably could be found somewhere in some data,
Right.
So you get all kinds of room to make all kinds of befuddling statements. But okay, molecular clocks. The basic concept of molecular clock is that under neo-Darwinian principle, or under neo-Darwinian thinking, where all organisms share common ancestry, that therefore our genomes come from that common ancestry.
Right.
And so the closer you are to a particular common ancestor, the more that certain parts of your genome should be the same. You all say, if you share a cytochrome gene, for example, with another organism, then that cytochrome from the common ancestor then will have changed over time as evolution's occurred. So when you look at organism A and organism B and you check their cytochrome, then that should be able to give you an idea of just how related they are and how distant they are from their common ancestor.
Okay.
So molecular clock is a way of going in and looking and seeing how much change genetically has occurred in this organism since it evolved from another particular organism. So that's the basic thinking.
Yeah, and right off the bat, we can see the assumptions of uniformity being tossed in there. The assumption being that these changes that are occurring and have always been occurring at a constant rate.
Right. And of course, they don't, and that's part of the problem with molecular clocks, is that they recognize, well, the changes aren't constant, because if nothing else, changes are based upon generation time, because you have X number of mutations per generation. So if organism A and organism B have totally different generation times, then that's going to dramatically affect the frequency, or that's going to dramatically affect the accuracy of the genetic clock. And so, yeah, the uniformitarian assumption is underlying the whole concept, and yet there's the recognition that it's not uniform. And so you're kind of assuming one thing, recognizing that's not the case, but still assuming it.
Right. Right. And in all of this, too, there seems to be this, again, as you said in your description, this flagrant circular reasoning here. They first claim that we have evolved. They then use the assumptions of who evolved from who. They then try to compare the organisms to figure out when they evolved from each other to figure out how long evolution has been going on for, which is then used as alleged evidence of evolution.
Right. Now, in fairness, if neo-Darwinism was actually true, and I was a neo-Darwinist, I'd be arguing the same thing, because that would be my assumption, is there's no reason to say that if organism A and organism B have a common ancestor 10 million years ago, that I shouldn't be able to then check genetically and see, okay, how much change has occurred in that 10 million years, and then use that to establish a genetic clock. One of the problems, though, is, in a sense, and one of the reasons it becomes a tautology, is I'm assuming, one, common ancestry, and two, I'm assuming a certain time frame. How do I know this particular gene is a better, or molecule, whichever way you're looking, either the sequence or the particular protein, okay? How do I know if it's a sufficient molecular clock or not? What is your calibration to know, does this one work or not? And the calibration is evolution. The calibration is, does it give me the 10 million years that the paleontologists are telling me I need? It does. Therefore, that's the molecular that I can use. That's the molecule, that's the gene that works. If it doesn't, if it says, oh, it really only says 1 million years between organism A and organism B and their common ancestor, obviously that doesn't work, so that molecule, that gene, doesn't work. So my calibration assumes evolution. And we see that just repeat throughout all of the molecular clock work, is you assume evolution, you see which marker works to fit the already preconceived idea of what evolution is, and then if it works, then we say, voila, we've proven evolution.
Sure. Yeah. Sure. Okay, now one of those clocks that I'm sure most of the viewers have heard of, certainly I've heard a lot about it, I've even talked about it on the show, we've heard lots about Mitochondrial Eve. Yeah. And in fact, as I mentioned it on the show. So this is a mitochondrial DNA molecular clock, correct?
Right.
So what's the story there?
Okay, the story is that all eukaryotic cells have a little organelle called a mitochondria. That's, of course, the site where most of the ATP is made. The mitochondria also happens to have a little bit of piece of DNA. Now, most of the time, a little controversy, but most of the time, you get the mitochondrial DNA from the female. So, for example, you and I both have our mom's mitochondrial DNA. She got that from her mom, et cetera, et cetera. Okay, so when you have that kind of lineage characteristic, then that already gives you the ability of being able to follow a specific strand of DNA back through particular family histories. Now, like any piece of DNA, the mitochondrial DNA is subject to mutation. In other words, every once in a while, a change may be introduced into it. Okay, so we can track these mutations and see, all right, how much change has occurred between your mitochondria and your great-great-great-grandma's mitochondria. And so by doing that, say we had in the late 1980s, some geneticists that did a study where they determined two things using mitochondrial DNA. They, one, determined that all contemporary humans have the mitochondrial DNA from one female, and that's why it became known as Mitochondrial Eve. Sure. And they named this female Eve not out of any kind of deference for even the Bible. It was a joke. It was tongue-in-cheek, making fun of. Yep. But of course, the fun thing of that is now they've introduced Eve into the scientific literature, so we can go into the public system and talk about Eve because it's in the public, it's in the public literature, it's in the scientific literature. So, that's right. God has a way of always working these things out. It's kind of amusing. Yep. So anyway, they identified what they determined was this single female, identified her as Mitochondrial Eve. Now, they weren't saying she was the only female alive at that time. They're just simply saying that she's the only one whose mitochondrial DNA has survived up to this point.
Right.
Everyone else, all the other females that were contemporary of hers, their mitochondrial DNA has died out. It hasn't been passed on. Okay, so that was one thing they found. But it is interesting how that is totally consistent with the biblical Eve, mother of all nations, mother of all humans.
Yep.
The second thing they did then was, as a typical molecular clock, they worked to determine, okay, just how long ago did this Eve live? In other words, we can take and genetically determine what's the distance between us and her in terms of years. Now, as the old saying goes, an interesting thing happened on the way to the forum. You know, interesting thing happened on the way to publishing.
Yep.
When they calculated mutation rate, they found out that it didn't give them the number that they wanted.
Right.
In fact, there's a paper in Science in 1998, almost 10 years after the publication of the original Mitochondrial Eve work, where there was the admission that if you use mitochondrial DNA based strictly upon its known mutation rate, in other words, on the data, you get an age for Eve of about 6,000 years.
How very convenient.
Of course, that is completely unacceptable. There's no way that's going to work, and no way you're going to get that published in the scientific literature. That would go over, literally the proverbial lead balloon.
Yes, yes.
So, enter in evolutionary thinking. See, because we got to think like an evolutionist, not a scientist. Think like an evolutionist. Okay. Evolutionists would think, all right, if the current rate of mutational change, the uniformitarian idea, is not working, it's not giving us the right age because it's giving us 6,000 years, and we know 6,000 years can't possibly be right, okay, then there must be a fudge factor necessary. So the fudge factor was, they calculated, okay, chimpanzees and humans have a common ancestor 6 million years ago. I think since then it's moved to 5 million years, but why quibble over a million years? Anyway, okay, so 6 million years ago. So what they did was they took the mitochondrial sequence of the chimpanzee and the mitochondrial sequence of the human, and they compared those and then extrapolated 6 million years and determined what kind of change that brought on.
Right.
And then they calculated mitochondrial Eve's age. See, and when you do that, then you get the reported 150,000 plus or minus years.
Right.
That's where that time came from. It has that evolutionary fudge factor in there. In other words, back to the tautology. If we assume evolution and we bring evolution into our calculation, in that evolution then gives us a fudge factor, so we can fudge the data to make it fit what we want, then voila, it works. We make it
voila.
What we want. And therefore, mitochondrial Eve lived 150,000 years ago.
Right.
If we go with the real data, what is the actual mutation rate? Well, it's only 6,000 years. Okay, well, obviously that can't be right. And so that's one of the things fascinating about mitochondrial DNA is that it supports very strongly the biblical view of Eve. Also, that's why you don't see very much written about mitochondrial Eve as a molecular clock anymore, because they lost it.
It doesn't work. Yeah.
And so more or less it was, well, obviously, and that's one of the things that the 98 paper in Science concluded. You can't use mitochondrial Eve as a molecular mark, as a molecular clock, because it doesn't work. Why doesn't it work? Because it gives us 6,000 years. So it obviously cannot work. See, what is our criteria for determining the accuracy of our clock? Does it work for evolution? Mitochondrial Eve did not work for evolution, so therefore the criteria says it must not work.
Right. And change the criteria then to make it work. Okay.
Yeah.
And now, of course, this may be a little too long to get into, but of course there is the Y-chromosome atom, as they called it, which was similar to the mitochondrial Eve, but just looking at the first man.
We guys didn't want to be left out. Exactly. Because you get your mitochondrial DNA most of the time from your mother.
Right.
You get Y chromosome obviously from the father. I mean, that there is no doubt about. There's no controversy in that. You have to get it from the father. So the idea is, okay, well, it works so well for mitochondrial Eve and for the women, to determine the mother of all people, let's use the Y chromosome and do the same thing as a molecular marker. Let's track the changes that have occurred in the Y chromosome through the males.
Right.
Now, an interesting thing again happened on the way to the forum. Just keep not getting there. What happens? Okay, we're looking at the changes that occurred in Y chromosome. The problem was that there wasn't enough changes that occurred. They couldn't find adequate changes. So the mutation rate was way too low to calculate, and therefore, because it was way too low to calculate, they did a very good job of saying that Y chromosome atom is 280,000 years, plus or minus 280,000 years.
Plus or minus what?
Plus or minus 280,000 years. In other words, because they couldn't find sufficient change in the Y chromosome, they just made up a number, 280,000 years.
Okay.
Oh, yeah. And one of the things they state in the paper is that they had to have a date that they felt was consistent with Eve.
Right.
I don't know why. I don't know why that was so worrying about them, but they had to have a date consistent with Eve. So 200, I guess, just out of thin air, 280,000 years. They don't explain where it comes from. It's just 280,000 years plus or minus 280,000 years.
Excellent.
Yeah, I wish I could have those reviewers. Man, what great work. Now, again, in fairness, there has been more detailed work done with Y-chromosome in the last 15 years, and he's gotten younger. Y-chromosome is younger now. Now he's not 6,000 years. I haven't gotten it down to that point yet, but last I saw, I think he was about 40 to 60,000 years old. So he keeps getting younger. Okay? But again, nothing has changed as far as the data is still not very accurate, as far as there's just not enough change occurring in the human Y chromosome to allow us to be able to determine a molecular clock very accurately to give us a Y chromosome atom. So they still continue to suggest then that obviously Y chromosome atom had to live Y tens of thousands of years ago, because that's what our evolutionary paradigm suggests.
Right.
The real data is we can't calculate.
Okay.
We can't calculate Y chromosome atom. It's just as likely as yesterday.
Right. Okay.
But you don't get that published. So
right, exactly. Yeah, who would publish that? Yeah, we don't know.
They just made up a fit with what they wanted, and that's how they get it published.
I see. Okay. Yeah. So now, speaking of molecular clocks, we know the decay rates of DNA, the deterioration of flesh, soft tissues, etc. And years before Dr. Schweitzer found soft tissues in a T-Rex bone, the Creation Research Society published soft tissues.
In 2001, Mark Armitage had reported the detection of collagen in a T-Rex fossil. Right. And that was 4 years before Dr. Schweitzer's first publication. But if we look in the literature, interestingly enough, we find that even Mark wasn't the first one. Other people reported it too. It's just Mary's is the one that's received the most attention. But it's been in the literature for a number of years. But it didn't make a big deal out of it. It was like, okay, this would appear to be, this could be, this might be. It does look like this is consistent with, they always hedge it with those kind of words. But we've seen then for quite a few years that there has been reference to what appeared to be soft tissue and what appeared to be proteins in dinosaur fossils.
Yes. Yeah. And of course, just recently, I'm assuming you caught this paper again. Dr. Schweitzer, just 1 or 2 months ago, finding now DNA in a hadrosaur bone.
Certainly finding evidence of. I guess the best way to put it is she used an antibody that had a binding affinity for certain regions on DNA.
Right.
And antibodies found binding sites. Even though she hasn't actually specifically chemically detected nucleotides.
Right.
She's detected molecules that these specific antibodies are binding to.
Right.
And she also used a stain that has a similar specificity.
Right.
And so she has been able to indirectly detect DNA by these antibodies and these stains finding sites to bind to.
And of course, they react with DNA. That's the whole point.
Right,
okay.
Right. And so that certainly is a precursor to that. There's obviously something there that is reacting as DNA would react.
Yes. So now my interest here, of course, is Creation Research Society had started up what they called the iDinosaur project, and I've sort of followed along with this. So what can you tell us about that?
You can go to our website, which would be www.creationresearch.org, and we do have on that website a somewhat detailed description of what we're doing, what we're trying to accomplish. Part of it really comes down to is we're not in any way questioning a lot of Dr. Schweitzer's work. In fact, she's really done some very good work. I know she gets upset when creationists talk about her work, but
she has. She's done excellent work.
Because it's certainly totally consistent with what we've been saying. And so how is it unfair for us to take data that's totally consistent with our position and use it? Where does that come from? She's accused us of rewriting the rules. You know, what rule is it that we can't use data that's consistent with our viewpoint? Our viewpoint is dinosaurs did not live 68 million years ago. And the fossils they're digging up were not 68, 70, 85 million years.
Right.
The presence of the tissue, the presence of the proteins, the presence potentially even now of DNA is consistent fully with that they're not 68, 70, 80 million years old, that they're just a few thousand years old. So why wouldn't we talk about that? I mean, it would be unscientific not to. So in that sense, regardless of what evolutionists may want to think are or not the rules, of course we can talk about it. Yeah, Dr. Schweitzer's done some very good work, but we just felt that it was appropriate for us as a research society and as a research facility in Arizona to obtain our own samples. We have the microscopic capability, we have some of the protein capability, we have some of the DNA capability. So it's appropriate for us to get our own samples, to do some of our own work, so that we're not dependent upon other labs to report or not report what they find, depending on if they want to or not want to. And so therefore, we're going to become a little bit autonomous and report our own work, report what we want to report as far as what we find, and not be worrying about, is this going to be accepted by the evolutionary establishment? Is this going to get me in trouble at my university if I publish this?
Sure, yeah.
I'm not accusing her of doing that. In no way am I accusing her of doing that. I'm just simply saying that we felt that it was totally appropriate for us to become independent, so that we now have control over what is done and what is published.
And it's replication of results anyway. It's proper science. Good science.
Replication, which any scientist should applaud. Let's replicate results. Isn't that part of the empiricism of science? Is that not only do you replicate results in your own lab, but other labs should be able to take and replicate results also.
Right. And
even
so
if we are those easy, wild, nuthead creationists.
Right. So now if people wanted to help out with that project, okay, funding obviously is one thing. Like, are you looking for dinosaur bone samples? I don't know.
We don't get funding from National Science Foundation or the Carnegie Institute or anything like that.
You don't?
Funding is, funding is strictly from individuals, churches, other organizations that support our work and want to help fund our research.
Okay.
So, and like any research, there's always money. I mean, money drives research. Research is expensive.
It is.
It's expensive equipment. It's expensive for time. Expensive for travel. We've had to travel to go collect samples. We pay other people for samples and such. So yeah, there's a nice money demand to it.
Right. Gotcha. Now, it's interesting. I hope you don't mind my going off on this rabbit trail here. Just this week, actually, we had booked this interview, and I get an email from a very prominent professor of genetics who wrote in. I won't mention his name because I didn't get a chance to get permission to read what he wrote. But basically, he was challenging creationary geneticists like yourself to submit your research to peer review. Now, I responded back to him saying, well, you guys have submitted it to peer review. You were only one creationary geneticist that came to my mind, but he was respectful, and I have no problem with his challenge. In fact, I want to be respectful in return. I'm not out to humiliate him or anything. But the thing is, as I pointed out to him, his conclusions that he was drawing, that he draws from his research, certainly were reproducible, testable, etc. But those conclusions were arrived at completely independent of evolutionary assumptions. Evolution is not a prerequisite for genetics research. And in fact, I said evolution had hindered scientific research. I gave just a couple of quick examples, like vestigial organs, the so-called junk DNA, which was assumed because of evolution.
Correct.
And now, behold, junk DNA has a purpose. Surprise, surprise.
It's not junk,
So I challenged him to dig deep, and he would see that his research in genetics can most certainly be done without any reference to evolution whatsoever. So in response, he provided one paper as an example of conclusions arrived at allegedly because of evolutionary assumptions and research. The paper was the P21 Cdk-Interacting protein CIP, CIPL, sorry.
The papers about some of the molecular mechanisms going on to control cell cycle and to initiate apoptosis if something goes wrong. And it's a very nice paper. I haven't read it in detail, but it's a very nice paper. But as you and I correspond a little bit, the word evolution doesn't even appear in the paper, and I don't see anywhere where you can see that by reading this paper, you think, oh, this paper is requiring evolution in order for me to draw these conclusions, or it's requiring evolution in order for me to be able to put this whole hypothesis together to test.
Yep.
I don't see that in there anyway.
Yeah, and I didn't either.
The best I was able to come up with is it's back to the idea of any and all changes evolutionists consider as evolution.
Okay.
It's a completely bogus idea, and they try to win by definition is what it comes down to. If it's change, it's evolution, therefore we win.
Right, gotcha.
So that's the best I can see is he's talking about these changes occurring in organisms and comparative, what's the P51 in this one and the cyclin activity in this one, and comparing it. And he would, I assume, be thinking that because of common ancestry, then that's what led me to do this type of comparison.
Sure.
Now, I'll give you an example similar to that. Several years ago, I published a paper where I developed the first platform for genetic transfer into anaerobic cellulolytic bacteria.
Oh.
My reasoning behind some of what I did was I was looking for genetically similar organisms that I thought would allow the transfer to occur better. Now, if I was an evolutionist and taking the position that this particular individual is taking, then I would probably be making the argument that evolution led me in that direction to think what I was thinking, because I think common ancestry and et cetera, and therefore they evolved from a similar pool, if you will, and so they're more likely to share genetic material. But of course, I'm not a geneticist, I'm a creationist, so I'm not an evolutionist, I'm a creationist, so therefore evolutionary thinking never even entered into how I arrived at my decision of what to do. So there's a classic example in my own personal research of the type of approach he would be claiming you can only accomplish if you follow evolution, and I did it, and I was not following evolution at all.
Right.
So they just impose onto what they do evolutionary ideas and say, see, it took evolution to figure this out. No, it didn't.
Right.
See, it takes evolution to understand this. No, it doesn't. But they impose that in, and then they pretend this makes it very important of how evolution is so important. Look how important evolution is, because you couldn't arrive at this if you didn't understand evolution. Yes, you would. The thinking I used in the comparative genetic comparison would be argued from an evolutionary perspective that this was requiring evolutionary thinking. But when I was doing that thinking, it wasn't an evolution doing that thinking. I'm a creationist, and therefore it didn't require evolution to think that way. Creationists can think that way too, because it's not a matter of thinking evolution.
Right.
It's not a matter of, oh, evolution's true, therefore this must be the case. It's a matter of thinking, okay, they're genetically similar, therefore. Because creation believes in nesting. And that's one of the things, if you understand nesting, the idea that you have groups of organisms that are very genetically similar. It's a nest. Creationists accept that. Evolutionists accept that. The idea is the nest rooted or not? We would say the nest is not rooted. Evolutionists say the nest is rooted.
Okay.
It's interesting that when you use molecular markers within the nest, they tend to work. When you get them outside the nest, they don't work. Well, that's kind of what creation would predict. Within the nest, you would expect the molecular marker to work. Outside the nest, doesn't work. Why? Because they're not rooted. The nests aren't connected. See, we don't believe in common ancestry.
Okay.
And so I would challenge again, and I would challenge your geneticist friend that's communicating with you, that this is exactly what the creation model predicts, is you're not going to get the molecular markers to work very well outside the nest.
Right.
And that's what they're finding. They don't work outside the nest. In fact, let me read you this real quick. This is out of the proceedings of the National Academy of Science last January, so January 2012.
Yep.
This is a paper written by Norman Pace. He says, quote, "It is now widely recognized that there can be wide compositional variations in the genomes of organisms that are normally classified together on the basis of their ribosomal RNA phylogeny." Okay? But that within group variation reflects the presence of cosmopolitan genes associated with more closely with a particular environment than with any particular organism. In other words, we're not seeing that these organisms that should be sharing genes are necessarily sharing genes. Who are sharing genes are the ones living in the same environment.
Right.
Evolution doesn't explain that very well. You got organism A and organism B. They don't share a very common ancestry. They don't share a very common nesting, but they have a similar gene. because of the environment they're in.
Right.
Right? And so what they're finding is they're having trouble following the phylogeny of organism A and organism B. And what they're having to say then is organism A and organism B acquired that particular gene independent of each other. They didn't acquire it from a common ancestor. They acquired it from, some horizontal transfer, for example. And so what we have is, then, as I say, the within group variation reflects the presence of cosmological or cosmopolitan genes associated more with the environment than with their phylogenetic history. And so that I find very fascinating, because that is exactly opposite of what neo-Darwinists were telling us 15 years ago. So neo-Darwinists 15 years ago were saying, look, we've got this nice, beautiful tree-like structure, as neo-Darwinism would predict. Now it's, okay, we have this bush, as neo-Darwinism would predict. No, so in other words, they're making the theory as plastic as they want it to be.
Right.
You get the tree, that's what we predicted. No tree, it's a bush, that's what we predicted.
Right.
You can't have it both ways. Did you predict it or not predict it? I had a biochemist friend back in the 1980s that was an evolutionist because of cytochrome C.
Right.
He referred to cytochrome C because of what he considered the evolutionary evidence from the sharedness of cytochrome C that evolution must be true. And talk all I want, he could not accept the idea that that wasn't legitimate, sound, absolute proof of evolution. Now, the irony is, with cytochrome C, we find that you don't have that nice hierarchical tree that he was claiming you had. Even back then, I tried to challenge him, it didn't occur, but he was convinced it was.
Right.
We find now with cytochrome C, for example, that, let me find a good paper here, with cytochrome C, we find that the turtle is closer to the bird than it is to a snake, which wouldn't make sense evolutionarily, and that we find that the snake is closer to human than it is to the turtle.
Right. That was the one I was familiar with. I was going to mention that, but
evolutionarily. So how did that occur? Okay, well, it didn't occur because of common ancestry. It apparently occurred because of all this horizontal transfer going on, or some other mechanism we don't know about.
Right.
But the idea being that this friend of mine back in the 1980s was a convinced evolutionist because of what was ultimately wrong. It was a molecular clock that was wrong. And how many other people have accepted evolution based upon some kind of evidence like this that was simply flat wrong?
Right.
How many people were convinced of the truth of evolution with the 1989 Mitochondrial Eve study?
Right.
And it was a beautiful study for creation.
Yep.
That's why we won't shut up. That's why we keep talking. That's why we keep bringing this out, because we are correcting these things.
Right.
And I had an evolutionist a few years ago tell me, what you said is totally true, but it's 10 years out of date. Now, I wonder if 10 years before he would have said it was totally true. It's funny how that, yeah. And so I'm sure now, if you heard me again, he'd say it's totally true, but it's 10 years out of date. I'm always 10 years behind. But I have a feeling that what I'm saying now, he wouldn't agree with, but 10 years from now, he'll say, I'm 10 years out of date. In other words, they're willing to admit we were right back
after the fact.
But can't be figured out. So now you're not right anymore.
Right. Gotcha. Okay. Now, did you not say at the beginning that you used to be an evolutionist?
No, I was never an evolutionist.
Oh, okay. I misheard you then. Okay.
Now, I started college as a creationist, but I would say I was probably a bit of a pliable creationist. In other words, if I would have really encountered some information, because I was that kind of wide-eyed, wet behind the ears, little student just ready to sop it up, because I was just anxious to learn, and I just wanted to learn, and becoming a scientist was my life's dream. I just wanted to get into that. And if I would have encountered what I would have considered, information that just right in your face, it would have been very, very challenging to me as a Christian and as a creationist. It would have been extremely challenging. And here's what happened. I took 2 evolution classes in college. Now, I didn't have to. My major professor, my major advisor was like, what are you taking these for? I said, well, I'm a creationist. I want to learn. And I called, you're a creationist. That was back at the time before you were considered, total slime. So he actually wanted to talk to me some about it. But so I took 2 evolution classes. And I sat there at the end of these were college level classes, and I got A's. Not because I'm trying to show how great a student I was, but just that I was paying attention. I was actually learning. I was actually, giving the answers and understanding the material the way the teacher was wanting. And I sat there at the end of the semester that I'd taken those 2 evolution classes and I looked at all my notes and I thought, this is it? This is it? This is all they have? This is evolution at the college level. Okay. This is a joke. This is an absolute joke.
Right.
I think those 2 evolution classes may be more of a creationist than any creationist information ever could have. Because I was done with those classes, I realized literally what creationists said all along. It is the emperor's new clothes. There's nothing to it. They come up with this idea, and you notice 10 years later that idea is gone. I call it the master magician approach. You know, the master magician, keep your eye here, keep your eye here, don't look back there. You know, the evolution is always, keep your eye here, look at this new evidence. Don't look at that evidence last year we got rid of that we were talking about and bragging about so much. You know, don't pay attention to that. Look right here at this new evidence. And they keep doing that over and over. As you point out, the junk DNA, the 1% myth between chimpanzee and human. You know, those are excellent examples. Mitochondrial Eve. We can go on and on. You know, the cytochrome C marker, go on and on of these that at one point in time were touted as wonderful genetic pieces of evidence for evolution. When the human genome first came out, I mean, the evolutionists were just giddy with themselves of just how beautiful this was as support of evolution. Just to tell you another story, I got all kinds of stories. I'll tell you another story. I was at a Gordon conference, and right before the publication of the human genome information, and I was talking to an individual who was actually involved in the project. So I won't give any more details. I want to identify who he is.
No names mentioned. Yeah.
That there were congressmen that were very concerned about the publication of this information. And why are they concerned? Oh, because it's just going to be devastating to those dumb fundamentalist Christian constituents of theirs. What? So I started pumping for information. Well, what is this? This is going to show. What's it going to talk about? And I went back to my room then that evening, and I wrote up a little article that was published in our little publication, Creation Matters.
Yep.
From this, I wrote the article, and I said, here's what the evolution is saying, and here's what I predict is going to be the case. Everything he said is no longer accepted. Everything I said has been verified true. And regarding the junk DNA, regarding the 1%, all of that, they were saying is going to be so devastating to these dumb fundamentalist Christians.
Mm-hmm.
It has all been debunked. It's all, they don't talk about it themselves anymore.
Right.
But everything, there isn't, you know, large amounts of junk. It isn't a 1% difference. You know, on and on. All that's verified truth. So my predictions based on creation have been verified. Their predictions based on evolution have been debunked. Now, which one is being more scientific? Which one is the better scientific approach? Which one explains the data better? Mm-hmm. It doesn't make evolutionists say creation is science or not. It's irrelevant. It's a moot point. Okay, not get into a word game, not going to allow them to win by definition. They can call it Charlie Brown if they want. The question is, does creation explain the data better? And the answer is resounding, absolutely it does. It explains the data far better. And therefore, if they want to say creation is not science, then they just have to acknowledge, okay, something that's not science explains the data better than their so-called science evolution.
Exactly. Yep. Okay, well, you're getting me started now too. I want to go off in different tangents, but we better call this quits. But thank you so much for being on the show today. I know for fact the viewers will really appreciate it. So thank you.
Well, thank you for having me.
This interview was edited for television, but you can catch the entire interview on our YouTube channel. Just head on over to genesisweek.com, which takes you to our YouTube channel. Stick around. We'll be right back after this break that is so short, it won't even show up on your molecular clock.